I have a couple of questions.
Okay.
All right, let's do it.
Okay, so when is test 2 going to be?
Okay, so that one's for me. So can you hear me?
Yeah, I can hear you.
Okay, great. So if you look at the syllabus, the revised syllabus schedule, it shows about when it'll be. Probably next week I will announce a hard date on exactly when it'll be. So if you want a straight answer right now, look at the syllabus and you'll get an idea of which week it'll be. That's all I can tell you right now.
Okay, and then another question I have is when will we be getting our Wa4 grades back?
So typically it's 2 weeks from the submission deadline. But the TA should be working on it right now. You guys want to chime in?
Yep, so WA4 is going to take a lot less time than WA3, thankfully, just because it's about 1 paragraph. So for each TA, it's probably going to be different, maybe by the end of this week.
Yeah, I told my class I'll have it done on Monday. We should have grades on Monday.
Okay, cool.
Yeah, I'll
Charles?
try for Monday. Yep.
All right.
There you
Looks
go.
like everybody's going for Monday.
You got your answer.
Let's see. Isaac asked if there's going to be any curve. Dr. Delafleur?
Curves to what? Specifically, like I can say.
Oh, that's a good question.
So
yeah.
I can answer in general terms. For example, quiz 3 was curved. So yeah, there's going to be curves. To Wa3, sure. I will, at the end of the semester, look at all of the grades for all of the writing assignments, and I will determine if a curve is needed or not.
Do you guys prefer questions asked over chat or over audio? You know, I think it's better to ask through chat, because if we have a lot of people here and multiple people speak at the same time through audio, we might get a little bit mixed up, but honestly, it doesn't matter. We haven't had that problem yet because we have only had like 30 people in our chat. So
well, right now we have twice what we had on Wednesday.
Oh, well, there we go.
So start asking questions.
Oh yeah, Cynthia, hey Cynthia, can we get some corrections / the right answer to the previous quiz and exam, please, so we can correct mistakes for the next exam? So for the exam, all of the material on WA1, it doesn't carry over. The understanding of it does, but you're going to be tested on completely new things. All the modules that we've gone through, so molecular genetics and bioinformatics, that's all going to be on exam 2, right? Basically everything after exam 1 is on exam 2. So you probably won't need exam 1. However, for the quizzes, Dr. Delafleur?
The quizzes are finished. There's no, so I mean, I hesitate, unless you want to do it on like if you want to Zoom with me specifically and we can go over, I can't just release the quizzes and the exams and give you guys the writing. I can't do that. So exam 2 is mostly going to focus on the bioinformatics and some general genetics. But the exam 2 is not cumulative necessarily. So you shouldn't have to worry about what you got wrong on exam 1, because it's not going to be repeated on exam 2, just outright. I hope that makes sense.
Okay, and then Isaac asked, so we just build upon WA3 for WA5. The broad answer is yes. We'll go into the nitty-gritty stuff shortly. How long should WA5 be? You know, 20 to 25 pages is probably going to be the average. The longer papers are going to get 30 pages, and then the shorter papers, which normally don't get the best grades, but there are some outliers, will be like 15 to 18, 18 to 20 probably.
So you're saying around 25 is including figures, including the pages.
Yeah, including the figures, including the figures.
So that includes that includes the cover page, the figures, and the references.
Mm-hmm. Will exam 2 have any outside reading that will be tested on exam?
Okay, so no, I'm already working on exam 2, and I'm gonna mainly focus on just the bioinformatics that you need to know. that you're working on now. I'm going to ask you questions about bioinformatics, but in order to work in bioinformatics, you need to understand DNA and proteins and their structures, things of that sort. So those type of general molecular biology questions that you should know by now as a senior or junior in a biology track, those might appear on the exam. There might be a few general fly genetics questions that you should already know. So, but no, I'm not going to ask you what Thomas Hunt Morgan said in his paper from 1920. I'm not going to do that. I hope that helps.
Will you be making the exam hard compared to quiz 3? Was quiz 3 hard?
So I did get quite a few emails of people that thought quiz 3 was difficult, and it shouldn't have been very difficult. That's why I had to curve it up. So, I mean, difficult is relative and subjective. What, you know, I had people that got 24s on exam 3 and people that didn't. So I don't know. It's going to be similar in difficulty to exam 1.
Okay.
So exam 1 was generally, again, with I think the mean grade for exam 1 was right in that sweet spot around 77, 76. So it did its job.
Good question. Good
So
question.
don't worry too much, because when it gets closer to exam 2, what I'll do is I'll put out a video lecture going over. I mean, I'm not going to give you the questions and the answers, but I'll give you some indication of what to worry about and what to study for exam 2. Anybody else?
Amount of questions?
The number of questions. That's a good. Right now I'm going with 50. Obviously, the more questions you have, the better you might do, because you have more opportunity. Like if you get a few wrong, you're still going to do better. It's going to be okay. But because we had to cut out a lot of the course. I mean, literally a third of the course disappeared. It may just out of necessity be a little shorter. Maybe 40 questions, 45 questions. So with so many people in the room, why aren't we not getting so many more questions?
Do we have a lot of people? Oh, we have 34. That's
35
nice.
people. That's twice the number of
Oh,
people.
what happened with Cinnabar?
So
Okay, so our Cinnabar mutant, the primer has an incorrect base, or the reverse primer specifically has an incorrect base. Those of you or those groups that have the Cinnabar mutation have been told about the incorrect base. What happened with it? I don't know. Somewhere along the line, that base changed from an A to a C or an A to a T, something like that.
So is that just I'm trying to find out, is that a problem with the sequencing?
No, it was just an edit, copy, edit, paste. Maybe somebody accidentally deleted something and then re-entered it on just transferring documents. Basically, the reverse primer was just slightly off, and so they weren't able to find it in their Word document whenever they searched for it. How specific should the bioinformatics section of the paper for Wa5 be? We'll talk all about it. What about Scarlet?
I don't think there's anything wrong with Scarlet.
There was nothing wrong with Scarlet.
So there's a question.
Difficulties finding one of the primers for Scarlet, you need to know what the nature of your lesion is for the mutation.
Yeah, so what that means is that you guys have a transposon. All right, and that primer is within the transposon. Okay, what is the time limit for exam 2?
So it's going to be probably about, since it's online and it's open book, you guys realize that, right? Because there's no way for an instructor to say, oh, no, you can't look at your notes. So it's an open book exam, just like quiz 3. So if it's 50 questions, you're looking at around 75 minutes. which is an hour and 15 minutes, which gives you about an hour and 10, 12 seconds per question. And you're going to be able to go back and correct. So you're going to be able to navigate your questions. So go back and forth. So I hope that helps. Yes, you'll have 24 hours to complete exam 2, but you only get one shot. You get one attempt. So the minute you start that timer, you have to finish. That timer will keep running until, and it'll automatically submit whatever you, wherever you're at 75 minutes. If I decide it's 75 minutes with 50 questions.
All right, guys, any more questions, comments, concerns? If not, we'll go ahead and move on to writing assignment 5. When is the group evaluation accessible?
I haven't deployed those yet. Those are now ready as of today. All the documents, everything's ready. So over the weekend, I'll probably send out emails. I'll put together a video on how to fill it out. So thank you for asking, but it's quite a bit of a job to get all of that information put together for everyone, but it's now ready. So over the weekend.
Okay, over the weekend. Any extra reading material for bioinformatics that we could be tested on?
What do you mean?
No, I mean, we have a lot of reading material out, reading and watching material, like videos that we've posted, guidelines for you to understand what the whole process means, what you're doing. As far as extra stuff though, like on exam 1, no.
Yeah,
for one of the figures.
Yeah, go ahead, Micah, go ahead.
For one of the figures in WA5, do we need to show the entire sequence with the highlighted Amplicon, or can we just show the Amplicon with the highlighted forward primer, reverse complement primer? Vinesh, that second idea is the perfect way to do it. Just showing the Amplicon with the forward primer, the reverse primer. Highlight them or denote them in some way, put them in capital letters. I don't know, whatever you want to do. Just make sure that we understand that, hey, this is the forward primer, this is the reverse. Good question.
Okay.
One of the group members is waiting to be let into the meeting by the host.
Okay, I'm sorry, that's my fault. I didn't look up and there's actually 2 people waiting. Sorry. Okay, 35 people. It's there's Shada coming in.
It looks like we have almost a completely different
Group.
set of people from last time, except for Danelis, Vinesh, and Samuel. That's
Okay,
good.
so if you guys notice that, because again, there's a limit to the size of the window. I can't see everything, so if somebody needs to be let in.
How many total figures do we need for bioinformatics? We'll go over that.
What's the short answer?
The short answer is 1, 2, 3, 4, 5.
Just for the bioinformatics?
Because you have the DNA sequence for the complement, DNA sequence for the mutant. You've got the Amplicon for the mutant, and then the Amplicon for the complement. However, some groups won't be able to get their complement or mutant depending on if it's transposon for the Amplicon there. Then so you also have, what was that?
I was going to say, what's the absolute minimum? Let's go from there. That would be
absolute
acceptable.
Minimum?
Yeah.
Okay. The absolute minimum, if you just happened to be one of those groups that had multiple transposons for both of your complement and your mutant, then you would have 3 figures.
Wait, hold up.
Or 4, probably 4.
Which figure is the DNA sequence?
Yeah, because go ahead, Charles.
No, I was just asking, Michael,
Yeah.
which figure are you referring to when you talk about the DNA sequence again?
So like the mutant compared to the wild type, so you can find the mutation in there.
That's one
Oh,
figure.
okay, the
That's-
Finch stuff then.
Yeah,
Yeah.
Okay, got
yeah,
it. Yeah.
Yeah.
Okay, cool.
because
Mhm.
I mean, I not to be contradictory, but I thought it was 2 to 3 figures minimum, and which is in the rubric. But we can go over that now, right? When we get to that part.
Mm-hmm. Well, the reason why I think you had the 2 to 3 was because it didn't mention putting the Amplicon in there as a figure.
Yeah. And then there's the optional structure of your protein, things of that nature.
Oh yeah, but I didn't count that one. Yeah.
Okay.
Let's see. Is it just me because your volume is suddenly very low?
Well, I haven't changed anything.
Yeah, I haven't changed anything either. So it's probably just you. How long must the bioinformatics? Okay, let's go ahead and start on WA5. Looks like everybody is shifting on over to those kinds of questions. I'll go ahead and start sharing my screen. Okay, there we go. You guys can see everything? Maybe?
Yep. I can see your.
Okay.
So where did my? Okay, we're almost to 40 people.
Nice.
So whenever you're ready, Micah, you can get started.
Yep. See, last time it made my little boxes go on their side, but I guess not this time. That's okay. Okay, Arusha, for Scarlet, since we can't find one of the primers, do we just highlight the forward primer and keep the rest of the sequence? You can, however, you need to tell us why. Why can you not find that reverse primer? And for those of you who have gotten to this point in the video, have the Scarlet gene and have no idea what I'm talking about, go back about 15 minutes because I told you what the Scarlet gene is and the reason why you aren't going to be able to see it. Okay, so title page. For the title, you'll have something that's going to identify your theme, study, or nature of the work. Right? On WA3 and all your other writing assignments actually where you've included a title, sometimes I left comments, sometimes I didn't. That doesn't mean that your title is perfect. It just means that I didn't comment on it. The best way to write these titles and what I'm going to be looking for when I'm grading them is a title that, one, has to include Drosophila melanogaster somewhere in the title to let me know what the model organism is. Two, you need to tell me what the importance of all of this is. What's the importance of your gene, your mutation, what you're studying. Three, you need to tell me what some kind of pathway information in that title as well. That's going to go along with the importance and also include the gene, right? So study of the mutant blank, study of the mutant Goldilocks, for example. One of my group's members has a gene name of Goldilocks. So study of the mutant Goldilocks in Drosophila melanogaster in relation to Hermansky-Pudlak syndrome. And I have no idea if that's the actual syndrome, but that could be an example title. The next thing is your name, institution, semester. Dr. De La Flor has provided a format for you guys on Blackboard. You just run through the entire thing. One thing about that is if it has like, I believe it actually has abstract 200, 250 words, approximately one page on the format, please delete everything besides abstract. It's kind of lazy if you don't. The other thing is, oh, for the formatting, he gives you sections, right? Make sure that you go in and put subsections into your paper. Okay, it'll help you guys organize your thoughts. It'll help you guys get a higher score, and it'll help us grade you and give you that higher score. because we're going to be able to see exactly where each piece of information should be. You'll get your scores back faster and they'll be higher. Oh, Vinesh asked if we would explain that in the figure. No, do not explain anything in the figure. Tell me what the figure is, right? You would explain it in either the results or discussion, right? You tell me what is in the figure and the results and then explain it in the discussion. Abstract. So when I'm grading you guys' abstracts, I'll be going a lot easier just because we haven't had a lot of face-to-face discussion. So whenever I'm grading WA4, it'll be easier than what I'm going to grade for the actual assignment. Right? I'll give you all the comments, everything you need to get a perfect grade for WA5, but I'm not going to grade as hard as I would on WA5 just because, honestly, you guys tried your best, you know? So in that abstract,
I was going to say, since we have a couple of the other TAs, Sarah and Charles, do you guys have any input on Micah's general approach? I mean, he's thinking of going maybe a little easier because we haven't had a lot of face-to-face instruction, or any since the break.
Yeah. So for me personally, I went pretty hard on WA3 so I can get in all those tough comments and, you know, give a little push for the students. So generally with WA4, and 4 is like medium, and then 5, I generally go easier, especially if I've seen you improve. But, you know, like I've sent out emails with pretty much every, you know, like with the outline of everything I'm looking. I even gave examples of, you know, kind of like an example of what I'm looking for in each section. Pretty much down to the sentence and down to the, at least down to the paragraph. So, you know, as long as you, for my students specifically, as long as you follow the outline well and you do all your homework and then you write it enough and everything looks neat, then you'll do fine. And as long as you improve, you know, over your previous paper. But, you know, some students, they had really good papers and I expect to give them pretty good scores at the end as long as they fill in the bioinformatics stuff. So, yep.
Yeah, I agree with that. As long as you improve on your WA3, take into consideration the comments that I left on the paper, and add in your bioinformatics, it should be pretty easy. I'm not going to be, you know, really hard grader, so.
Thank you. It's all yours, Micah.
So the abstract should be 200 to 250 words, and you need to have a word count at the end, in parentheses.
Should I make this into Michael?
So who's that?
That's Michael. Was that a question to us, or was that just background noise? That was probably background noise.
Yeah, I don't see.
Okay. Isaac asked if there were any samples of WA5 on Blackboard.
No.
Mike?
I can't, no, I'm not going to put, I mean, other than things like sample figures, there are no examples of, say, a good WA5. Just because what ends up happening if there's one like that, you guys have heard me say this before, then everybody's paper looks like that one good WA5, right? So we can't do that. So that's why we're doing this Q&A. And we did one on Wednesdays to talk about it, go through the rubric, answer questions to get you guys where you need to be.
And you can also always email your TA. I've told my students that I'm not going to revise their papers like I have been for all the other ones, because all those revisions before now should have prepared you for WA5. However, I will answer literally any question you have. So if you send me 100 questions, I'm going to answer 100 questions. I'm just not going to go over a 25-page paper.
So the Q&A from Wednesday will not be uploaded because we had technical problems with that recording. It's one of the reasons that we're having a second one. So
yes,
just happens sometimes.
Yep, and there will be other Q&A sessions as we get further along, probably whenever you guys are almost done or close to being finished with WA5, just to see where you guys are at, see if you have any questions. And okay, let's keep on going. So the abstract needs to describe the biological context and rationale. So you need to mention that you're using Drosophila melanogaster somewhere in that abstract, right? You should mention the importance somewhere in the abstract as well, right? So that importance from the title, what your gene is related to, what the study is related to in humans, mention that in the abstract. You should go over your aims or objectives, which is basically what are you trying to do with this study. Briefly mention the methods. Don't give us class-specific information, like we performed wild type cross 1 and wild type cross 2, DBM1, DBM2, because in a real paper, those names mean nothing, right? What you should provide to us is what those crosses were for. So we performed a cross with double balancers or double balancer flies to assess the inheritance pattern of our mutant fly, and we found this. Boom. So you've just told me brief mention of the methods and the result from that method. Okay. And then the brief mention of the overall conclusions of everything. And the abstract could stand alone from the whole paper, which means that you shouldn't have anything that I would have to go into the paper and figure out, like, okay, so they mentioned this in the abstract. I need to go through the paper and try to figure out what this actually means. Okay. Can we get penalized for having a paper that's too long? Tashfia and all of my students, I don't care if your paper is like 35 pages. Honestly, it doesn't need to be anything more than that. Like if you were absolutely like, that's the longest it should probably be.
Yeah.
It would be a miracle if you're able to get it to 40 pages. I had one person write me a beautiful introduction filled with literally anything and everything under the sun. It was 5 pages long, but you don't need that much information to get a 100. Okay. And at a certain point, especially in the materials and methods, more is actually going to hurt you.
Yeah. So yeah, I want to reiterate that while I'm not going to step in and keep someone from writing a 35-page paper, it's really not necessary, especially since we removed all of the molecular work. Earlier, Micah mentioned that a good paper would be about 25 pages long, including the figure pages, the references, the cover page. So if you could keep it to about that, that would be great. Now, as Micah said, if you really want to make it 35 and you really have that much to say, then sure, I guess that's fine.
Yeah, for my students, I personally prefer a more in-depth introduction. So if you go a little longer in the introduction, I'm happy with that, because that's the interesting part. Methods, the shorter the better, and then for results and discussion, it's kind of whatever. So yep.
Kind of whatever. Okay.
Yeah. Well, and then especially like this semester, we had a lot of students who didn't get to certain crosses. So if you're comparing your paper to your friend's paper and your friend has like 5 more pages than you and you're wondering where those pages are, it's probably because you didn't get there. Okay. And you're not going to lose those points as long as you communicated with your TA that those points shouldn't be on there in the first place.
So I did mention on several emails and on video that you need to tell your TAs where you stopped your fly work so that they know how to grade your paper. I'm sure most of you have already done that, but I want to reiterate that, and Micah just did the same thing.
What similarity percentage do you guys expect? I honestly don't care about similarity until it becomes a problem. All right.
Yeah. Okay,
Okay. But if you know it's going to be a problem, then don't make it a problem. Okay. So it just means don't cheat.
Yeah. So to have a more sort of quantitative answer to that, 15 to 20% originality score, or similarity, I think is what it is, similarity. Any higher than, especially on Wa5, you start creeping into the 30s and 40s, and that's not good. You don't want to do that. And if you notice that, because you can see your similarity score, if you notice that the similarity is coming from one source, like a paper from someone else, I'm not saying that you would do that. We can see that as well. And that's a clear indication that something shady is going on.
Yeah.
If a lot of the paper is being lifted out of another paper.
Yeah. So if you have a paper that is over 20% and you didn't cheat, don't worry about it. Okay. I'm not going to care because I read all these papers. I remember all of the papers. So I'm going to know exactly who you're copying off of. Don't worry about it. Okay. And if I can't remember a paper from a previous semester, then turn it in and we'll remember it for me. Okay. Introduction. Introduction should be 2 to 3 pages. Like Charles said, the deeper you go, the better here. However, there is a certain point where you, in my opinion, that you should probably stop off. And that's going to be like right at the end of 3 pages for me. You should give us a broad overview of D. melanogaster. One point that I noticed on WA3 that a lot of people missed was they told me that D. melanogaster was great because it was small, it was cheap, had a 2-week life cycle, and all the little bitty things that we tell you in the very first day. But then they didn't go deeper and tell me how it's been used in science, and that's where you guys, almost all of you, lost points there. So I want you to go a little bit deeper and don't just give me the surface level stuff about DML whenever you're talking about it in your paper. The next thing is states broad aims of the study in words or why you are doing this work. This should just be like a topic sentence. This one, in my opinion, should probably go towards the end whenever you are talking about your major conclusions. So you start off with the broad aims of the study, and then you go into your conclusions, and everything before that is introductory. Introduce candidate gene and how it's discovered or how a possible mutant was identified. Discuss at least 2 other related genes. This is not your WA1. A lot of people unfortunately pasted their WA1 into the introduction for WA5 or WA3, sorry. That was not how to do it. You should have talked about your mutant gene, specifically the pathway behind that mutant gene.
So you, I mean, if you want to take it down with Jonathan.
What?
Ask him if not, I'll go.
Hello?
Hey, if you guys aren't talking, can you mute your stuff, please?
We good to continue? Okay. So let's see, you've got your mutant gene, right? Talk about that, talk about the protein pathway behind it. Then you have your 2 other related genes, okay? Those 2 related genes should come from that protein pathway, or they should be related to the protein pathway of that gene in some way. And you just have to tell me that way. For genes like yellow, those of you who cannot find 2 other related genes, you're gonna have to extrapolate a little bit. Get creative. Talk about your protein or your gene in the sense of another protein, how it interacts with another protein, and talk about that pathway, okay? And the genes related with that. Some of you will have a really easy time writing this section, and some of you are going to struggle a little bit. It's going to be okay. If you have any questions, email me. Reviews previous literature as part of the justification of the study, why you should discuss the biological processes appropriate for your gene. This one is kind of wrapped up in the previous one. If you've done the previous one well, you'll get these points easily. Right, because that previous introducing the 2 genes, the pathways behind them, and your mutant gene, you're going to have to do a lot of research there. You're going to have to pull a lot of information from other papers. You'll get the points just by doing that. Elaborate on the basic biological molecular processes involved with the mutant gene function. Again, same thing as we said previously. And then after this, you should have a small conclusions paragraph. Right? Not the same thing that you put at the end of your conclusion, but just something that mentions the major results of the paper, right? So you should go through the crosses, you should go through the bioinformatics, and then the big overall result of that, boom, done. Okay, any questions on that? Nope, okay. Materials and methods, 3 to 4 pages. Honestly, the shorter, the better with the materials and methods. You do need to describe how the flies are cultured and manipulated. And you have to include all of the materials that you use, right? So you use the brushes, you use the microscopes, you use the fly buddy. You used a camera, possibly. You use, let's see, the vials, the food, and incubators. A lot of you forgot the 18-degree incubator. Make sure that you remember that, because I'm sure that all of you have used it, or at least the people that I've spoken with in my class. Lists the fly strains used and explains how the crosses were conducted. The most successful way I saw this done on WA3 was dividing it up cross by cross by cross. Each cross had a different section. However, I did have people successfully get a full score on this section if it was just one long paragraph. However, it's a little bit hard to do just because of the organization point there. Describe how relevant equipment and materials were used. Okay, most of you got this except for those who forgot the incubator. Describes the blast and bioinformatics analyses. You should talk about the cutoffs here, right? So for the e-value cutoff for nucleotides, it's 10 to the -6. For proteins, it's 10 to the -3. And then tell me what kinds of blast you use, so P-blast and N-blast. It shouldn't be very long. You should be able to do it nice, short and sweet. And you also need to talk about statistical analyses. I don't know if I mentioned that, but do talk about chi-squared. The things that I want to see on chi-squared are what was the test used. You actually have to name the test. All right, you need to tell me the p-value you used, you need to tell me how you calculate degrees of freedom, and you tell me the conditions for rejecting or failing to reject your null hypothesis. Those are 4 things. All right, so remember those 4 things in a nice, brief section, and you're good. A lot of you lost points for protocol. I just wrote protocol on it and crossed off a whole bunch of lines, and your paper was a sea of red. If you saw that, that just means that you are speaking too much, right? Your materials and message should be short and sweet, and you should never, ever, ever give me any reasoning on why you're doing something, right? So your crosses, you shouldn't tell me that you're doing the wild type reciprocal crosses to do this or the DBM cross to do that. You shouldn't tell me that. You just tell me that we did the DBM1 cross this way. Boom, done. Oh, the other thing on this, you need to describe the phenotypes. Whenever we have the scoring or how the flies were scored, you need to actually go through and tell me how you identified each phenotype, right? So curly have curly flies, Daiki has wings out at the 45-degree angle, Plum has darkish brown eyes. Let's see.
I'm answering
good.
in text, so I'll take care of that one.
Oh, okay, awesome.
So I'm going to write a little more to Isaac.
Okay. Let's go on to the results then. The results are going to vary between groups and those writing the results. Why? Because some groups had more information, some groups had less information. However, no matter what, you should have something written for every single cross. Right, you should at least have the scientific basis for every single cross, even if you don't have the fly progeny, the numbers, the counts, the figures that go along with it in like the actual pictures. You should at least have the scientific basis, okay? And then for DBM2, you should at least have the scientific basis and the chromosomal diagrams. And as we mentioned in our last live stream for WA3, those chromosomal diagrams can come in one of 2 forms. They can come with a picture. You actually draw out the little chromosome. I saw some really, really awesome ones. Great job, those who did it. And then I also saw some really awesome ones of the way that Dr. Delafour suggested last time with just writing out the genotypes. Either way, you're going to get the full points as long as you do them correctly. Okay, so for the results, this is all stuff from the previous paper. All of the comments that I've left on the previous papers and that your other TAs have left on the previous papers should be applied to WA5. And if we see improvement, then you'll most likely increase your score from that section, right? Good.
So just one thing, Mike, I wanted to reiterate that blue indicates what I thought should be a required figure, but we can change that here and edit the rubric, and I'll upload it again. if you want to send me a copy.
Okay, so this right here describes mutant phenotype. A lot of you put this in the introduction section in my class, or not a lot, but some of you put this in the introduction section. This is a result, put it in the results section, and then you should have that figure from WA1 to go along with it. Describes genetic basis for rationale for WT1 and 2. You got the chromosomal diagrams, and then, or if you need them for the sex links, right? And describes genetic basis and rationale for deviant1, deviant2, and wild type cross. You got chromosomal outcomes here, chi-squared tables here, and all of your tables and figures, everything in the figures section should have a figure legend. Every single one. I know it's a lot of writing, but they all need figure legends. You can't just put them there. Complementation test crosses. Honestly, I didn't see a single group. Actually, there was one group that had the complementation figures, like with the pictures, and that's great. Awesome job. But if you don't have the complementation pictures, you don't need to put it in there. It's okay. Bioinformatics results. Okay. Describes the results. Sorry.
So, Micah, if I could interject, they still, if you could also, if they're missing an experiment, whatever that experiment is, they still need to write about what it would have been.
Yes.
Right?
Yeah.
So they still need to write.
So you need to tell me the scientific background on it and the expected results from that experiment. Okay. We're good there?
Yeah. So yeah, that's great. So you need to understand what the purpose of the experiment was, the rationale, what you might have expected to see from that experiment, and what it means.
Yes. Okay, so bioinformatics is going to describe the results of blast analysis of the DNA sequence. It will identify the allele of the unknown mutant gene, and you must show the DNA sequence alignment data in a figure. Okay, so what does this mean? Let's break it down real quick. So describes the results of the blast analysis of the DNA sequence. Those results are going to include an S value and an E value. You need to tell me those. The other thing is you need to tell me where the mutation is, or where you think the mutation is, right? For those of you who are unable to identify the mutation or have no idea what you're looking at, all of the mutation information is on your allele page in Flybase. It'll tell you exactly where the mutation is, exactly where it is, okay? You have to show the DNA sequence alignment data in a figure. And I think I'll just run through that one more time, just so everybody knows what DNA sequence alignment is.
So yeah, somebody just asked, I think it was Danelis, she asked, I think it's a she, I'm sorry.
It is.
Can you show how to do the alignment? So you're about to do that? Is that what you're about to do?
Yes, okay. I am about to do that.
Are you laughing at me, Sarah? Is that what you're doing?
No.
It's okay. That's okay. We should also, I'd like to, while Micah is getting this ready, I know this is a lot of work and it's made more difficult because we can't meet face-to-face and have that personal instruction. But I know this may just go in one ear and out the other, but try to have a little fun with it and not take it too seriously. Like, don't freak out. Don't worry too much. You'll probably end up doing a better job if you don't worry too much, and just stay in touch with your TAs and with me, and we'll get you through the other end. We'll do the very best for everybody. Trying to put people at ease. It's not the end of the world.
Okay. Here we go. Can you guys see this?
I still see Wa5.
Oh, really?
The Wa5 rubric, I mean.
Ah. Well.
It says I'm viewing Micah's screen and I see Wa5.
Let's see.
Now I see your camp.
Okay. All right, what about now?
Micah has started sharing. Yes, I see a Flybase.
Perfect. Okay, so I'm going to show you exactly what you need for that DNA alignment figure. Okay, so let's say we're doing DNA alignment of yellow. Cool. We're going to yellow. We'll look it up on Flybase. We're waiting, relaxing, maybe drinking some coffee, some water. Eventually, we get a search result. I'm not sure if this is Flybase or my internet. All right. So I'm just going in, I'm getting the sequence of yellow. Wow, this is taking a long time. Okay, new plan. I'm going to just show you what it looks like on one that I've done previously.
Is everything okay?
Yeah,
Okay.
I don't know if it's either my internet or my computer, but I'm going really slow right now. Okay, can you guys see this? Everything on my
Yes.
screen?
Yeah, I can see it.
Okay. Okay, so this sequence alignment right here is going to be like what you'll pull up for me in your figure legend. Okay, you should include everything from the top line right here to the bottom right here. I should be able to see the score, the e-value, the identities, the gaps, the strand plus minus, all of that, right? You should be able to identify your mutation somewhere within this right here. Identify that in the results, talk about it in the discussion, and then in the figure legend itself, tell me how you identified it, right? So, hey, I highlighted it, or I denoted it with an arrow or something, okay? All right, so that's the figure, right? You need to give me one for the mutant and then one for your complement. All right, you also need to give me the amplicons for each as well. So those Finch TV figures, or those Finch TV sequences we gave you, or no, I don't know. Yeah, the Finch TV sequences we gave you are going to be used to find the blast between the wild type and those Finch TV sequences, right? And then you'll use your primers to give us the amplicon. So theoretically, it would be 2 figures for the mutant and 2 figures for the complement. Some of you will not be able to get the amplicon, and so you won't be able to show us 2 figures for each, and that's okay. Okay, describe, what's up?
Nothing. I'm going to be in and out AFK every 10, 15 minutes. Sorry.
Oh yeah, no problem, no problem. Okay. So next point is describe the molecular basis and results of blast analysis of genes protein sequence when examined for matches in the human or other species database. Okay, some of you will not have a human match. That's okay. If you don't have a human match, let us know and talk about other species. Some of you will have human matches. That's great. Talk about both. All right, talk about both the humans and the other species. You need to talk about any conserved sequence domains, and preferably tell us what those sequence domains are used for, right? So if it's the active site of the protein, or if it's a alpha helix, beta sheet, something. Just tell us information about any of those. You must show the amino acid sequence alignment data in the figure. Okay, this is the easy part. All you have to do is go on Flybase. You can do it one of two ways. You can get your DNA sequence and translate it to a protein, or you can just get the protein sequence straight off of Flybase. There's an option to do that. And you take that protein sequence to BLAST. You'll blast it, and you'll be able to find the similarities between a human homolog or homologs in other species. All right. And then you also need to talk about the E values and S values there as well. The next one is refers to the appropriate figure or table within text, presents all results in a narrative and logical flow objectively with very brief interpretation. All figures and tables have a proper legend. Okay, this is important. You should always, if you have a figure somewhere at the end of your paper, you need to mention it somewhere in the paper itself, right? Don't just leave it hanging. Because when I'm going through, I'm checking to see like, okay, so table 1, I go down to table 1, table 2, go down to table 2, table 4, where's table 3, right? So make sure that you include or you reference all of your figures in the paper somewhere. Oh, there's questions. Let's see, Danellis asked, if we don't know got to get there, we cannot do the complement. I'm not sure what that means. Could you ask that question one more time? I'm sorry. Which of the given Finch sequence are we supposed to use? There is 8 in my folder. Yes, Sin. There is going to be a lot in people's folders. You should use the one that has your gene. Like if you are Sin, you had yellow, I think, right? I'm pretty sure you had yellow. Yours will have like Y, a couple spaces, Y, F, yeah. So like Y, YF, Y, YR, right? So yellow, yellow forward, yellow, yellow reverse. And then it'll have another one for your complement gene. Whatever your complement gene was, it'll have the complement gene, complement forward, complement gene, complement reverse. Those are going to be the ones that you're going to focus on for the wild type versus mutant comparison. Will there be a video on how to get the Amplicon? I think that's what Charles did, right?
Yeah, so go ahead, Charles, go ahead.
Yes, right now I'm rendering the higher quality version of the video. I've already uploaded one copy onto the OneDrive for like the full video, and I covered the so I started off with the Amplicon stuff, and then I went to the Finch TV stuff, and at the end I cropped in my protein blast stuff. So it should be everything in one video.
Okay,
so
awesome.
As soon as that's done, I'll post it to Blackboard, a link, and then Micah, you can grab it and put it on YouTube as well.
Yes.
Yep.
Please, one more time, give a final run through of bioinformatics figures we need. Okay, good question. You need the DNA sequence alignments of both your complement and your wild type. You need Amplicons of both your complement and your wild type if you're able to get them. You need the protein alignments of your protein or your gene's protein versus human protein, and your gene's protein versus another organism's protein. If you're unable to find human protein, just give us the other organism's protein. And also, if you want to talk a little bit more about that other organism and how that gene plays a role in the other organism, and you want to include a picture of that other organism, you can do so. Because I mean, I like looking at cool pictures. Okay, we find a human homolog for a gene, right? Not just our mutant allele. Sorry if this is not clear. Yes, you need to find a human homolog for your gene. if we don't know how to get the alignment part.
Again, that Charles just finished saying that he's putting up a video that will cover all of that step by step.
Danellis, Charles just put a video up on it. If you still don't understand it, I will personally FaceTime you or Zoom you, and we can go through this step by step. Because you have been working hard on this section. I promise it's painful, but at the end, it's going to be worth it. Okay, discussion, 4 to 5 pages. Some of you unfortunately missed the discussion mark on WA3, and it was kind of sad, but it's okay. It's okay. Your discussion should cover all of your results. It should not be 1 paragraph long. You need to tell me what all of your results mean. If you had to reject a null hypothesis, you should address that in the discussion, okay? Go through every single result. Some of you did amazing on the discussion and actually saved your grade because of the in-depth nature of your discussion. Let's see. So for the discussion, you need to explain why your candidate genes hypothesized in WA1 were wrong, or elaborate on why 1 was correct. Go through your process of elimination for your candidate genes here. A lot of you forgot that on WA3. However, some of you did remember that one. Let's see. And here, whenever we say to tell us how you eliminated them, I need you to go step by step. In this cross, we found out that our gene was sex-linked, right? So you crossed off all the autosomal genes. In this cross, we figured out our gene was on chromosome 2. So you crossed off all the other genes that were not on chromosome 2. In this cross, we found out that our gene was this, which is a complementation cross, right? And so we were able to cross out all these other genes, which either means that we were left with 1 gene that is we hypothesized correctly with, or we don't have any of the genes, and that's okay. You don't lose points for guessing incorrectly. Explains observed genetic interactions between unknown gene, or if genes are and other genes appropriate for your mutant experiments. A lot of you will not have any of the genetic interactions. For those of you that do, though, go ahead and take a crack at it. You got this. If you'd like to add any interactions between your gene and another gene that was not discussed in this class, go ahead. That's a great time to do it. The discussion is the time whenever you give me all the extraneous information that I wouldn't expect to see anywhere else besides your paper. Addresses experimental limitations and possible sources of error. Do not tell me that we killed all the flies because somebody in my group left them on the fly pad for too long. All you need to do is tell me, let's see, like we had limitations. We had problems with stocks, right? So some of your stocks just did not want to have sex. You did not get new progeny. That was unfortunate. That's okay. Tell me about it. All right. Tell me about literally any other issue besides I messed up. Bioinformatics limitations. We discussed in the last session that there really was not any bioinformatics limitations. But Mike, what did you say about this? I forget.
Oops, sorry, I'm doing a million things here. In regards to what,
okay.
like let's see.
It said bioinformatics limitations.
Oh, yeah, so I put that in there in case there were any problems with missing primers, or primers that aren't working, or sequences that are messed up, or something like that.
That would be good for those of you with the Cinnabar gene.
Mm-hmm. So you could write about any issues that you encountered.
Okay, okay. So explains how sex-linked versus autosomal genes differ. This should be an entire paragraph all by itself, and get into the genetics on this, all right? If you get into the genetics level on this, you get the points. If you just tell me that sex-linked is on the X chromosome and autosomal is literally on the 2, 3, 4, and that's it, and it's 1 sentence, you will not get the points. Compares results to those of other investigations, published sources about your allele. So look up your gene, and especially that first paper that discovered this gene, and compare your results. All right. did you get that your gene was on this chromosome? How did they see, or what were their reciprocal crosses like? What were the results from their crosses, and how do they compare with yours? So this is almost like a confirmatory paragraph saying that, hey, my results are true. I've had people in the past get the same results. The next one is explains how mutations in the gene can give rise to a mutant phenotype at the molecular level. For example, how is the protein product different in the mutant and wild type? Okay, this one you kind of have to walk through a little bit, just briefly, the central dogma of molecular biology. So like gene, DNA to RNA to protein, right? And when you have a lesion in the DNA, your protein is going to change in some way. And because of your research into this gene, you know exactly what changes, right? So is it a change in the active site? Are you deleting an entire portion of the protein and it's no longer functional? It just gets chopped up as soon as it's produced by the ribosomes. Let us know. Okay, so walk us through that process. Walk us through what happens at the protein level whenever you have this mutation. And then compare the differences between the wild type and the mutant, right? Especially if you guys have a mutation in the active site, there's a lot that can be said there. Okay, I mentioned Wednesday, if you want to go into the electrophysics, getting some physical chemistry in there, I'm ready for it. Hit me. Okay. Oh, and how does that difference give rise to the mutant phenotype? So this one, you're going to kind of go back to your introduction because you should have discussed this a little bit in your introduction on what's actually happening in that protein pathway whenever you have a mutation in the gene. All right, so if you have an ABC transporter, something's not getting transported. Okay, tell me what it is. Tell me where in the process it's not getting transported. Let me know what's happening. Indicates importance of experimental findings with possible applications and compares the melanogaster gene to similar genes in other organisms. Okay, this is, in my opinion, the meat of where it's all at, right? Just because it's telling me why your research is important. Okay, everybody loves flies, or for the most part in science, everybody loves flies. But what people really love is to stay alive. All right, so we're using these flies to figure out how we're gonna fix other people that are sick, okay, in the long run. So tell us the actual application for this research, right? Compares DML gene to similar genes in other organisms. Okay, so you're gonna go kind of over your blast results here. You're getting into more blast here. Talk about humans, talk about other organisms. I would honestly prefer both, but if you really can't find humans, it's okay. I know some of you will not be able to. Discusses demelanogaster gene product protein relative to at least one non-Drosophila homologous protein that matches well from a blast analysis. So this is going to be either human or another organism. Talk about that protein as much as you can. For those of you who have a human protein, you're going to be able to give me a lot more information than those of you with a, or rather, you're going to be able to give me a lot more information a lot easier than those of you with a protein from, I don't know, a bug of some sort. Let's see. Okay, proposing new questions for future research. All right. So this one has 3 points in my view. For a future research question, this one has 3 points. It should be a section all by itself, by the way. The what, the how, and the why. All right, all you need to do is give me 2 of those points to get the full grade on this, and then you're done. You walk away, you never think about this again. All right, you need to tell me what you're going to do. That's the easiest one. All right, all you have to tell me is what you're going to do in the future. Why you're going to do it. That's the second easiest one, right? I guess a step above the easiest. Okay, so why are you going to do this research? And the third one is how you're going to do this research. Whenever people give me all 3 of them, I get really, really happy. Okay, really happy. So if those of you who can get all 3 successfully, that's great. I've had like maybe 5 people in the past do it successfully. They give me an amazing like 3-point future research section. So good, so good. And then the last thing you should do is summarize major conclusions. A lot of you forgot this section or just kind of tacked it on as one sentence at the end of the previous paper. Make sure that this is its own paragraph, all right? So you summarize your major conclusions and you provide an adequate interpretation of all experimental results. That should occur throughout the discussion. So this right here, if this is on the rubric, this is kind of like the did you mess up anywhere? All right. This is more for the TAs to separate the really good from the perfect. Let's see, literature cited, 1 to 2 pages. If you have a good introduction, you don't need to worry about the literature cited because the literature cited is going to write itself as long as you have a good introduction. If you cannot get up to 11 to 14 peer-reviewed references, there's an issue. All right, it means you got to look more. Make sure that all of your references are correctly formatted. Dr. Delafleur really threw you guys a bone on WA3 because he made this section worth an entire letter grade. So for those of you who had great references sections or literature cited sections, awesome. You literally got 10 points right there for doing nothing, especially those of you who use some kind of reference software. But for those of you who kind of did not do so well on the literature cited, you need to really make sure that it is correct in WA5. For the future section, does that have to be after discussion or just at the end of discussion? It should be towards the end of the discussion. The end of the discussion should be the conclusion statement, like the conclusion paragraph, just brief at the end. Okay. Use proper reference format. Overall general formatting. Parenthetical citations are required throughout the paper. Make sure that you are using in-text citations correctly. I had some people who were not. Proper font, spacing, margins, justification. Make sure all that's correct. I was pretty easy on, honestly, I was pretty freaking easy on the spacing and the margins and all the little bitty things for formatting on WA3. Just because of COVID, all the stuff happening. I know there's a lot of stress trying to figure out what you're doing with your lives now that we're literally stuck at home all day. So honestly, I did not take off a lot for formatting for WA3. WA5 is going to be different because I commented on everything for you guys. All right, so just make sure you remember left-right justification. Genes are going to be italicized. Proteins are not italicized. Drosophila melanogaster is italicized. D. melanogaster is italicized. The figure legend should always go under the figure. Your paper should run from paper to literature cited to figures. Paper, literature cited, figures. How long would you prefer the future reference section to be? Honestly, Vinesh, I don't care. I've had people get it like full points on it, remember, with just the what and the why. They had like 3 or 4 sentences, boom, done. Then I had somebody give me a page and literally like, I thought they were about to write a dissertation for me. Okay, it was beautiful. They obviously got the 3 points and like the full credit for the section, and I was a little bit happier, so I may have just like scooted the grade up a little bit on a couple other things. But honestly, it doesn't, I don't really have a preference on that.
So this is just a section for you to think about how what you've learned about your gene could possibly be applied to future research questions. Be creative. As long as it's biologically plausible.
Yes.
Just write about it.
Yes. Yes. So I've had people try to shoot for this section to get the 3 points, and I normally see it one of 2 ways. 1, it's really well done, or 2, it's so far away that I'm questioning whether or not it's science. So you don't have to go through for all 3. You can just give me the what and the why and be done with it. You get a 100 and move on with your life.
Michael?
But if, yes.
Just a quick question. Did you mention the superscripts?
Oh, Charles, thank you so much. Okay. Okay. So for general formatting, your alleles should always be superscripted. Don't give me the caret. Don't put the 1 next to it. Don't put a space.
The little box.
Yeah, please
Just
don't put a
superscript.
space between them either. It should be like, here, let me show you where the superscript is. I've found that a lot of people don't actually know where the superscript is. And honestly, when I found out that this button existed, it became my favorite button because I could write like chemical equations. And back when I was like very young, I thought that was super cool. So look, H, look at this, subscript 2, and then O, boom. Okay, beautiful, right? And then if you want to write an allele, we do yellow 1, superscript, boom. Okay, well, it changed the yellow because of that, but it doesn't matter. Anyways, remember to use the superscript and subscript buttons, please. It's a button right here. There's also a shortcut on your keyboard. For those of you who first world probs, okay, we're about to do some word 101 right here. First world problems. I don't want to move my cursor to use the italics button. Control I will automatically put you in italics. Control U will automatically put you in underlines. Control B will automatically put you in bold. Okay, for Macs, it's Command I, Command B, Command U. Is there a shortcut for superscripting? Yes, there is. I don't know what it is for Mac.
It's Control Shift Plus. Yeah. Control
Shift Plus. Okay, there we go. All right. Appropriate genetic and taxonomic nomenclature, and then appropriate grammar, sentence structure, avoids jargon. All right, so this one, for those of you who have English as a second language, you started off the class and I absolutely destroyed your writing assignment 1 because of grammar. All of you have really improved, and I'm incredibly happy on that. You did a great job fixing and revising and asking me for help and coming to me with questions. It was great. It was amazing, and I've definitely seen a positive trend in the grammar and sentence structure of your papers. Awesome job, guys. There are some people still struggling. The times when I'm going to take off for this is when it hinders my understanding of what you're actually saying, okay? And it's not just English as a second language people, it's Americans too who have lived here their entire lives and speak English, okay? You need to really, really focus on sentence structure, sentence flow, and thought flow. Okay, don't just write something and then jump off to another topic. You need to transition to it. All right. Make sure that your thoughts are coherent. Make sure that you're completing them. And please have somebody go over your paper for grammar. Those of you who have thrown grammar out the window, please go and get it. Take it back inside. It's cold out there. Okay. Please improve your grammar, because it's only going to hurt you if I can't understand what you're saying. All right. Well, there we go.
All right, so we had some good questions there for 10, 15 minutes, and then Micah just went over the Wa5 rubric with some input from the other TAs as well. So anybody have any other questions? Okay, so I don't see too many other questions being asked. Well, there we go.
I struggled to find human homologs that I felt comfortable using for my paper. My gene is white though. What am I doing wrong?
So which?
Okay, your gene is white.
So there should, go ahead. I'll let you handle it.
Okay, so you should definitely find some human homologs in there. 100%.
Right.
100% you should find human homologs. That is an ABC transporter. It's been extensively studied, and you actually should write a very solid introduction. In my opinion, that gene, the white gene, the scarlet gene, are probably some of the easiest to write the introduction on, just because of how much work has been done on them already. So what do you mean by that I felt comfortable using for my paper? I'm not exactly sure what you meant by that. How about singed? I couldn't find 2.
So Micah?
Yes.
if you want to do this, you can screen share and go to Flybase, and there's actually an human ortholog section right underneath in Flybase. So if you want to just screen share and show them real quick.
I would love to do that. However, my computer is like actually frozen.
Okay. Let's see if I can do that.
Okay, so while Charles is doing that, it says, how about Singed? I couldn't find one either. Yes, Singed is one that's probably going to be difficult to find a human homolog for, I believe. You should definitely be able to find some other homologs in other species, though. Valeria, do you search for ABC transporters instead? No, what you can do is just blast that gene, and it should come up, like, blast the protein sequence of the gene. Okay, blast the protein sequence, and you should be able to find a human homolog of the ABC transporter in there.
Is my screen sharing, by the way?
Yes, yes, we can see your screen.
Okay, so let's just do white. Let's try white. Wow, it is slow.
Yeah, because I also brought up Flybase, and I was going to try to do something with it, but I noticed that it was and I'm at U of H, and it was being a little slow.
Interesting. Okay, go to your gene, white, and then on the right side, there's a part called orthologs.
And then human disease associations right there.
Yeah, exactly. And there it is, all your ABC transporters. So ABC, so you just look at the, let's see, best score. So this one's the one with the highest homology score, but all of these all probably have a decent E-value. So all your ABC transporters. So there you go. And then we'll do one more. So the other one's what, Singed?
Sure.
Yep.
Yeah, I remember one of the groups, I forgot what it was, they didn't have a human homolog, but I think most of them do. So there it is, orthologs, and then human, yep, HSAP, and then,
oh,
there you go. Yep.
All right, that's good, that's good.
Yep. Okay, stop share.
Okay.
Valeria, is what I meant by not comfortable is that the papers seem to say that it's inconclusive. It's a little bit interesting. However, Charles just went through finding those homologs for white, so I think you should be covered there.
So, okay, good. Give it a shot. So stay in touch with your TA. We may do this again because we like doing it. So, okay, anyway.
All righty. Any more questions?
So
how many people do we have right now?
So we had a high of about 40. We still have 33.
Wow. We're doing pretty good.
So we'll leave it open for a couple more minutes for questions. If we don't get any more questions, we'll call it a day, and we'll very likely do this again whenever we decide to do it.
Okay.
Any other questions? Okay.
Oh, please upload this video. Yes, we will upload it ASAP.
Yes, we will upload this video. Last time we said, like I said, we had a technical glitch and we weren't able to do it. I blame Micah's cat. Okay. All right. Well, any last words from our TAs? Sarah, Charles, Micah, anybody?
No. Yeah, I think the updated version of the video will be done in 3 hours, and then I'll upload that on OneDrive, and then it should all be good. And if you guys want to take a look at it, at least the TAs, or if you want to take a look at it, Mike, I already have one version of it up.
Okay.
But it's just super blurry. So you'll have a better version.
Yeah.
In a bit.
So it's the same thing with Zoom. Once we're finished here, I click end meeting, and then it begins to render the video. And depending on how long it is, it can take up to an hour just to render the MP4.
Okay.
All right. Okay, so it's now, okay, there's another question.
This isn't really a question for WA5, but for the final, are we still going to the university to take it?
No.
What?
No. There's no final. There's exam 2, which is not cumulative, and it'll be online. This is for Michael. Just like quiz 3. Okay. Yep, you're welcome. All right, so I think I'm going to officially adjourn this meeting, and if the gods smile upon us when I click end meeting, it will begin to render. Okay. So can you see, Shaida, you're saying, can you see your quiz? Sure. If you want to send me an email and you want to go over the quiz, we can do something like Zoom like this to go over what you missed. But it's really, I mean, what's on the exam is going to be different. So if you're trying to somehow hedge your bets that way, it's really not going to make any difference. Okay. All right. It's 4:30. I think an hour and a half is long enough. But are we done?
Yep.
Okay.
Should be good.
We're done. I'm going to click end meeting, and well, actually, I'm going to click stop recording and then end meeting. All right. Hopefully it'll work. Please say a little prayer. Bye-bye.
See ya.
Bye, guys. Have a good day.